Hello, welcome to video 1 of 2 on protein purification. Protein purification is an essential process within the field of biochemistry. If you're already familiar with expression, you should know our proteins synthesized by the expression vector are produced along with other natural proteins and byproducts of bacterial cells. The goal is to sift through these impurities and eventually get to a pure sample we can study and characterize. Today we will be learning about affinity chromatography, an essential type of purification among many others. If you want more background regarding purification, I suggest starting you off on these videos. Without further ado, let's begin our journey into affinity chromatography. Following expression, we have collected a whole cell lysate. Our protein is trapped within the cell, so we use various methods to lyse the cell and release all of the soluble proteins into the extracellular fluid. This is done chemically with lysozyme or mechanically using sonication or freestyle methods. DNase is then added to break down large strands of DNA. Next, we transfer the lysate into a centrifuge tube and spin down the lysate, separating its contents into a pellet and supernatant. The supernatant contains a majority of proteins we have just released from the cell. We transfer the supernatant out of the tube, then filter it to get rid of any loose debris and large tangles of DNA not digested by the DNase. We then load the prepared sample into the purification column. The sample loaded into the column is now referred to as the crude, and the fluid running out of the column is the flow-through. Taking the crude and flow-through and running it on an SDS page should show an empty band on the flow-through when comparing it to the crude. This simply demonstrates that our protein has successfully bonded to the column while the majority of bacterial proteins have been washed out. The selective binding is no fluke, but rather some exceptional planning at the cloning step. Your expression vector was designed to include a tag portion at the end of your protein, which bonds strongly to the column matrix. Demonstrated in this diagram, we can see the histidine residues in a histidine tag bind very well to nickel ligands embedded in the column matrix. Now we have reached the actual purification step. Here we release the protein from our column and collect its contents in a process called elution. Comparing the structure of histidine to imidazole, you can see the two are very similar and both bind very well to nickel. Because of this, introducing imidazole to the column competitively binds to the nickel ligands, releasing the previously bound proteins. Buffer A, the binding buffer, contains little to no imidazole compared to buffer B, the elution buffer, which contains a high concentration of imidazole. Mixing the two together creates a controlled gradient of imidazole, which we can use to accurately wash out the column of any bound proteins. Elution and mixing buffers is controlled by the FPLC, or Fast Protein Liquid Chromatography machine. The FPLC I have just introduced to you has another useful function. With the help of some software, the UV lamp and detector reads the absorbance of the sample flowing out of the column, which is then recorded as a chromatogram. The absorbance is a good indicator of protein concentration because of the high absorbance of tryptophan, a common amino acid found in protein chains. Naturally, a high UV absorbance would be a sign of high protein concentration. Here's a chromatogram I have taken the liberty of cleaning up. As you can see, the y-axis measures UV absorbance, while the x-axis indicates the volume of fluid passed. The blue line tracking the absorbance paints a picture with many peaks, signifying certain points in the experiment in which high concentrations of protein was eluded. Meanwhile, the green line is set at an arbitrary height to identify what percent mixture was used. Note that there are multiple increasing flat curves on the green line. This is a standard washing method called step gradient. As you can see, each time we increase the imidazole concentration, an absorbance peak follows shortly after. Once the peak tapers off and plateaus, the column has eluded all proteins, with a certain binding strength according to the imidazole concentration. Also note that the new baseline is formed as buffer B concentration increases, since imidazole has absorbance as well. The final relevant feature of the FPLC we will be talking about is its ability to collect the sample in a process called fractionation. Simply put, a junction changes the route of your sample to flow into tubes ready for collection. The tubes, known as fractions, fill with sample according to trends we see on a chromatogram. This way, when each fraction corresponding to a peak is run on an STS page, we get an accurate representation of what each peak contains. This concludes our lesson on affinity chromatography. The concepts you have learned today are applicable to many more types of purifications you may encounter. Once you are familiar with these concepts, please watch video 2, where we put these procedures into practice.